codon. Okay? Or should I say, one amino acid can be specified by more than one codon. When we say the genetic code, which is the language of instructions that the protein machinery is going to use to interpret the information on the RNA, is degenerate or sometimes redundancy. What this means is that for one amino acid, it can be coded for or specified by more than one codon. An example in the slide, for example, the amino acid alanine has more than one codon. For example, GCU will still give you alanine. GCA will still give you alanine. So just for one amino acid, you've got 4 codons. Also, another example is leucine. Leucine also has got more than one codon. When an amino acid can be coded for or specified by one codon, we say that nature of the genetic code is degenerate or redundancy. But now, it's important for us to understand that not all codons exhibit degeneracy. No. For example, the codon for methionine is not degenerate, meaning that AUG will just give you methionine, or methionine, the amino acid, can only be obtained from one codon, which is AUG. The question is, how does the machinery allow for one amino acid to be specified by more than one codon? This brings us to this property of Wabo base pairing. Here, we want to understand how is it possible that one amino acid, like alanine, can have more than one codon specifying it. I want us to look at the codon. Yesterday, we said we can number the codon as having the first position, second position, and the third position. What you see in this slide is leucine. And here we can see that we've got our mRNA, and what is bound to it is a tRNA. We're going to look at tRNAs very soon. In this slide, what we just want to focus on is the codon that is on the mRNA. In the other picture also, we can see this is our mRNA, and these are the codons. If you look at position 1 of this codon, we have a C. Again, when you look at position 1 of the other codon, again you have a C. So we can see that the first position is the same. We can do the same and compare the second position. You have a uracil at this point, and also at the second position, you also have a uracil. So far, the two codons are the same at the first and second position. Let's go to the third position of the codon on the mRNA. On this codon, we have C. On this codon, we have AU. We can see that while the first two positions are the same, the third position is different, but both of them are going to give us leucine. What this means is that leucine can be obtained from this codon, CUC, you still get leucine, and also when you have CUU, you still get leucine. But the property of Wabo base pairing is in reference to the third position on the mRNA. It appears that this position can bind loosely to the tRNA. Of course, this is when we go to translation. It is not fixed. Now, like I said, not all codons, not all amino acids have got more than one codon. For example, we talked about methionine. So Wabo base pairing, this is when the codon on the mRNA at the third position can mismatch, but allow for the entry or for the selection of the same amino acid. Okay, now let's go to another property of the genetic code. The genetic code is continuous. It is not punctuated. Normally, when you're reading or when you're writing, we use punctuation to give meaning to what we are saying. And also, to rest, as you read, there's no way you can read a paragraph with 2,000 words without a full stop, without a comma, and things like that. So normally our language in English, in Bemba, and all those other languages, we use punctuations. But the language of the genetic code is not punctuated. Sometimes It's even referred to as commaless. They're not commas, they're not full stops. What does this mean? Let's look at the mRNA that I've designated with an A. We can assume this is a 5' end. And from yesterday's lecture, we understand our mRNA, we are always reading it from the 5' going to the 3'. And how are we reading it? We're reading it in codons. A codon is just a sequence of 3 nucleotides. So if I stand at this point, if you can see my cursor, I can start by reading AUG. This is going to be 1 codon. The moment the ribosome is done with reading this codon, it will not stop. It must continue to read the next codon. In this case, the next codon is going to be CAU, and it will continue all the way until the end of this mRNA. This is the right way of reading the genetic code. And this is what we're referring to as a genetic code is continuous, not punctuated, and also not commaless. Let's look at B. This is another mRNA. If it were punctuated, what we'd expect is that after reading AUG, what you see here is a dash, maybe the machinery should stop and start reading this again independently. So what you see in B and in C where you have commas, it will mean that the machinery as it is translating this codon is going to be interrupted because of these commas or the dashes. In short, on this slide, the take-home message is that one of the features of the genetic code is that it is continuous, it is not punctuated, or should I say it is not commaless. In addition to that, one of the features of the genetic code is that it is ambiguous. Unambiguous. What this means is that each codon only specifies one specific amino acid. While we can have more than one amino acid being specified for by more than one codon, but each codon should only mean one thing. So we're saying that the genetic code is ambiguous. Each codon, okay, in the genetic code specifies only one amino acid. For example, in the slide, when you have AUG, this should only give us methionine. CAU, this should only give us histidine, and so on and so forth. When we say the genetic code is unambiguous, what you're trying to say here is that each codon only has one meaning, and that meaning in this case is in reference to an amino acid. So at this point, we need to remind ourselves, yes, we're saying that one amino acid can be coded for or specified by more than one codon, but the codons that specify this amino acid can only mean one thing at a time and not two and not three. That is what we're trying to say here. And another interesting thing about the genetic code is that it is universal. What you see in the slide here, you can see a number of creatures. Most of us have seen some of them, some of us have not. But of course, if at home you have a dog or you have a cat or a chicken, there's no way in your mind you can think that the instructions that allow your cells to function, the instructions that, in this case, when I say instructions, we know, I'm talking about this, the genetic instructions, right? There's no way you're going to think that they could be different. But guess what? The instructions, and remember the alphabet is just an A, a C, a G, and a U. They're the same, it's the same alphabet that the cat next to you at home, the dog, the chicken, if there's a snake on your farm, and all those, we're using the same alphabet. So what we're trying to say here is a genetic code is universal. These organisms that we see around us are using the same information. Now, the question that one should ask is that, but then why do I look different from the chicken I'm going to have for lunch? Okay, if you're going to have beef, and if you can, just imagine the face of a cow, there's just no way that the cow and me should have the same alphabet. But then why are we different? At this point, we've established the cat in your house, the instructions that it is using for life, and your instructions are the same. But the question is, why are we different? I know my cousins from Chipata, they always laugh at the Bembas and talk about monkeys and the stuff. In fact, as we go on to look at gene expression, you're going to see that, between a chimpanzee and a human being, We almost have a 99%, genome identity. But why are we different? Just look at the pictures that I've shown you. If you can see this cat is very good. I would have put, well, there's a donkey. I would have put a cow. Or maybe look at this zebra. Sorry. What animal is this? The one with the long neck. Can you think of yourself, having the same genetic instructions as any one of these? And if not, why are we different? This is a question that I'm posing to you. Why don't we look like a tortoise? This is a tortoise here. That's a giraffe there. There's a penguin there, and so on and so forth. Look at this ostrich. Looking very funny, but why don't we look like them if all of us are using the same genetic instructions? Someone to give it a try. So just unmute yourself and you can speak. So guys, the question we're asking is very simple here. We're saying that the genetic code, the instructions that the cell utilizes to retrieve information from mRNA so that we can get our proteins. And our proteins, we know, they determine so many things. They're involved in so many processes. The structure of our cells, enzymes, there are so many things that proteins do for us. But then at this point, we're saying that these instructions, they're the same in you and in me, and the chicken we're going to have at lunch. So when you go for lunch today, when you look at your chicken, just know that the instructions in the chicken and the instructions of you who's eating it are the same. The question is, why don't we look like chickens? Why don't we look like cats? But anyway, these are some of the things that we need to look at as we learn or just think about, because really getting this information just for passing is good, but also just to think beyond. Okay, and it also helps us understand other topics that we're going to look at in future, such as gene expression. So since no one wants to answer, I'll just continue. So now the answer is that, or should I say, part of the reason why we don't look like the chicken that we're going to eat is because while we have the same instructions, okay, the way these instructions are written or expressed in this case is different. You can think of the alphabet. You have 26 letters. And if right now I asked you to describe what you have done up to this time, assuming this is, let's say, 11:10, we're going to use the same 26 letters, and all of us are going to have different descriptions of what we have done, meaning the information is going to be different. That's assuming all of us write different things. You can think of it the same way. We only have 4 letters in the genetic code, but how these letters are combined can give us different information. How these codons are placed can give us different information. In yesterday's lecture, one of the things that we noted was that the sequence of nucleotides on the mRNA, and in this case now we know that the sequence of those codons determines the polypeptide sequence. And also it's going to determine or influence how the protein is going to fold, and finally how the protein is going to function. So while we are using the same genetic information, how this information is shuffled, in this case how this information is laid out and expressed, is going to be different. And that's how come the chicken we're going to have for lunch and ourselves are going to look extremely different. In addition to that, one of the features of the genetic code is that while it does not have any commas, any punctuation of some sort, it has signals. We know that it has signals that allow the machinery to know that this is where you start, that is a start codon, and it also has signals within the mRNA that tell the protein machinery to say this is where translation should stop. So you can think of it like when you're writing a sentence. Normally, we start with a capital letter. So when you see a full stop and then a capital letter, you know this is where the sentence starts. So even when you're reading, we know where the sentence should start. We also know where the sentence should stop. That is at the full stop. So think of it this way. The genetic code then also has, in its own way, some form of punctuation. That is to say, it has signals that guide the protein machinery to say, this is where the information on the mRNA I'm carrying should start, And we know the start codon is an AUG. It also has signals that allow it to know that this is where this information should end. And some of those, and we know there are 3 of those stop signals. That is UAG, UAA, or UGA. Those ones, I remember yesterday telling you to say, you must know them. At least we should know the start codon. We should also know the 3 stop codons. But to memorize a genetic code, that will be too much information and very unnecessary. So up to this point, we are done with the first part of translation. So now I want to move on to the second part, which is protein synthesis. So by moving to protein synthesis, we have understood how this information on the mRNA is going to be read. That is to say, we understand what a codon is. We also understand the direction in which we're going to be reading this information. We know we must always stand at the 5' end of the mRNA and we move sequentially towards the 3', reading these nucleotides in 3 or in codons. Now we want to find out how is this information now translated into a protein. We're going to look at this process in 3 stages. That is not to say these are not all the stages. Remember, we have to pick the main ones. The first one we're going to look at is amino acid activation. And just by the word amino acid, at least you know part of that word is that we're going to be looking at amino acids. Then we're going to go on to initiation of translation, elongation, and finally termination. What are our objectives in this part of the lecture, the second part of translation? I'm hoping that by the end of this topic, we'll be able to explain amino acid activation, or should I just say tRNA charging. And in this topic, or in this subtopic, we're going to look at how amino acids are selected from the cytoplasm and brought to the ribosome. Because the understanding here is that for translation to complete, with an understanding that the mRNA is carrying different codons, and each of those codons is specifying an amino acid, it means that in the cytoplasm, you must have a pool of amino acids. But then how does a cell select which amino acid should be brought to the ribosome to be added to the polypeptide chain? So it starts with amino acid activation, or should I say tRNA charging. Then the next thing now, we're going to look at how translation, initiates. How does the ribosome know that this is the start codon? Okay, that is what this part is going to be about. And then we're going to move on to elongation, that is just addition of more amino acids, the formation of the peptide bond. From our amino acid lectures previously, we looked at amino acids as the building blocks, and to build a protein, we know these amino acids have to be linked by those peptide bonds. We're going to see how are these peptide bonds formed in the ribosome. And then now we're going to move on to termination. In termination, you want to understand how does a protein synthesizing machinery know when to stop? To say, the information I was reading, now I've approached a full stop. I'm not talking about the full stop in the actual sense of a sentence. I mean, this is a stop codon, so I should end here. And finally, overall, we expect that at the end of this lecture, you're able to explain how translation converts the information that is encoded in the mRNA into a functional protein. Understand the whole picture. So I'll start with amino acid activation. What is amino acid activation? Or in this case, sometimes it is referred to as tRNA charging. This is just a process by which a specific amino acid is selected from the cytoplasm where it's existing in a pool of other amino acids, and it is covalently attached to the tRNA before translation begins. What this means is that before we can even initiate translation, something has to be done. And that something that has to be done starts with selecting the right, or should I say, the correct amino acid. So before we look into how this amino acid is selected, we need to understand who is selecting this amino acid. These amino acids are selected, or should I say, we need an adapter. that is going to enable us to select this amino acid and bring it to the ribosome for protein synthesis. So it's important for us, first of all, to understand the structure of tRNA. At this point, we understand tRNA is transfer RNA. Transfer RNA is an adapter that links the amino acid in the cytoplasm to the information that is on the codon on the mRNA. Now let's think of an adapter. Right now, if you have your phone, if you charged your phone, the adapter, what did you do to charge your phone? You place your socket on the adapter, your plug of the adapter on the socket to power it where there's a source of electricity. And on one end of the adapter, you got a cable and put it to your phone. So the adapter is a link between the source of electricity and your phone that you want to charge. So let's think of that scenario in line with what tRNA is doing. So on one end, tRNA should bind the amino acid, and on one end, it must bind the codon on the mRNA. So it's acting like an adapter. That is why tRNA sometimes is referred to as an adapter. So to understand how it is functioning as an adapter, we also need to understand its structure. Generally speaking, the tRNA is said to have a clover leaf structure. What you're seeing in the slide is referred to as a clover leaf structure. Like any other mRNA, like any other RNA molecule, it also has an orientation. You can see on one end it has a 5' end, and on one end it also has a 3' end. This is very important, and we know why. Because when you look at DNA replication, transcription, the direction in which these molecules occur has meaning in the way, for example, they're duplicated or read out. In a similar way, it's important for us to appreciate that tRNA also has an orientation. It has a 5' end on one strand, on one end of a strand, and the 3' end on the other end. Now, in this clover leaf structure, we have what is referred to as the acceptor arm on the 3' end. The acceptor arm on the 3' end, this is where the amino acid is going to be attached. The amino acid will be selected from a pool of other amino acids in the cytoplasm, and it's going to bind to one end of your tRNA. Which end is that? Is it the 3'? Is it the 5'? We are saying that this amino acid, which is selected from the pool of other amino acids, binds to the 3' end. Guys, there's someone who has their mic on. I'm going to request that you mute your mic unless you want to say something. But also, I'm going to ask that don't interrupt him. Like I told you, my laptop, or should I say my battery, is quite low, so I'm hoping that we can cover as much. We have a lecture tomorrow morning. If you have questions, we can still pick them up tomorrow. The person who has the microphone on, kindly mute it. Is it Herbert? Kindly mute your mic. Thank you. So what was I saying? I was saying that the amino acid will be selected from the pool of other amino acids in the cytoplasm, and it's going to be covalently attached to the 3' end of the tRNA. This region on the tRNA is marked by a specific sequence, CCA. And this is a sequence that allows the amino acid, of course, we're going to look at how it is added, to be attached or to recognize which end it should bind to. In addition to that, what you see on this tRNA, we have a D arm on this end. We also have a T arm on this end. These arms of the tRNA, they help in the selection of the amino acid that is going to be bound to the tRNA. They also help in the folding of this tRNA molecule. Most importantly now, I want to draw your attention to this end of the tRNA. At this point, we're saying that overall the structure is a clover leaf. It's got many other features. But I want us to focus now at the anticodon arm. Okay? What we're seeing here is that this is a region of the tRNA that is going to bind or complementary base pair with the codon on the mRNA. This region of the tRNA is referred to as the anticodon arm because it has an anticodon. Now, for now, let's say I take out this, the beginning of this word, I take out the anti. I just remain with codon. And remember from the mRNA, what a codon is just a sequence of 3 nucleotides that carry information, one, either to give us an amino acid, or they can be a translational sequence such as a stop, start, and things like that. So when we say the tRNA has a codon, it means it also has a sequence of 3 nucleotides in this arm. This sequence of nucleotides, which we're calling a codon, is referred to as anticodon. Now, anti means that it is complementary to the codon on the mRNA. So in this regard, I hope I can explain that better in the next slide. We're trying to say that the tRNA has an anticodon arm. The codon in this word just refers to a sequence of 3 nucleotides. Okay, and anti means that this sequence of 3 nucleotides is complementary to the codon on the mRNA. Remember, the mRNA is carrying different codons. I just want us to hold on to that thought. So what this means is that you're going to have different tRNAs for different codons. At this point, we're trying to say the relationship between the mRNA carrying our codons and the tRNA is that the tRNA carries an anticodon that is complementary to a specific codon on your mRNA. Some of the features I think I explained in these notes, and in your own time you can go and expand them. Okay, let's look at the relationship between the tRNA, which we can see here, and the mRNA. The first thing that I'm going to start with, I want us to pay attention to the orientation of our strands in the tRNA. We said the 3' end, this is the acceptor arm. It's going to accept the amino acid to be covalently linked to it. And it's got that marker sequence. Okay. It also has a 5' end at the other end of the strand. The portion we want to focus on next is the anticodon arm. We said the anticodon is complementary to a codon that is on the mRNA. Now, remember I said pay attention to the orientation of the tRNA. This is a 3' end of the tRNA. This is my mRNA. You can see on this end of my mRNA, this is the 5' strand. Now, if I move closer to my codon, it means this end is a 5' end and this end of the codon is the 3' end. The anticodon then, this end is going to be a 3' end, this end of the anticodon will be a 5' end. This is very important because translation also proceeds in a specific direction. Okay, so now let's look at the anticodon-codon relationship. That is to say, the tRNA is carrying the anticodon and the mRNA is carrying our codon. I'll start by standing at the 5' end, the first nucleotide. On my mRNA, I have A. On the anticodon, I should have the complement. Remember, this is RNA, we don't have thymine. That is why at this position, we have uracil. On the second position, it is U. It complementary base pairs with A, which is adenine, and so on and so forth. What you see here is that the anticodon is complementary to the codon on the mRNA. What this means is that if you have another codon here, it means, and it is different from this one, we need to have another tRNA to come in to read that information. So meaning that you need to understand that the tRNA has a relationship with the mRNA in the sense that it complementary base pairs with the codon using its anticodon arm. And what this also means is that this codon here should specify methionine. AUG gives us methionine in this case. What this tRNA should allow to bind to its acceptor arm must be methionine. That is why I said the tRNA is like an adapter. On one end, it must bind the amino acid. On one end, it must establish complementary base pairing with the codon. Now, if this codon is methionine, what this tRNA should be carrying is methionine. If this codon was leucine, should be translated to leucine, the anticodon must match, and indeed, what this tRNA should be carrying should be leucine. So then, the tRNA is an adapter, allowing for the machinery to select the correct amino acid that is complementary to what is presenting on the anticodon codon, position of the tRNA. That is to say, there can be no match here if this amino acid is wrong. It means that the anticodon will not complementary base pair. If this is not enough, you can go. I've added a slide which notes for you to go on and just understand that process a little bit more. Now, at this position, should I say at this point, we have understood the relationship between tRNA and RNA. We understand that tRNA is just an adapter, linking the amino acid, bringing the amino acid, the correct amino acid that is being specified by the codon on the mRNA. Now, charging of the amino acid, that is bringing it from the cytoplasm to come and bind to acceptor arm, is a stepwise process. Okay, the charging of an amino acid, that is to select it and bring it to the tRNA before it can bind to the codon of the mRNA, is a stepwise process. So the first thing that happens is that an enzyme that is referred to as amino acyl tRNA synthetase is going to bind the amino acid. This binding of the amino acid is guided by what is presenting on the codon of the mRNA. Remember, I told you, for there to be complementary base pairing between the tRNA and the mRNA, the anticodon-codon must match. So meaning that when this enzyme, amino acid tRNA synthetase, selects the amino acids from the cytoplasm, it means that the tRNA already has established what is sitting on the codon in the ribosome. In short, the enzyme that helps the protein machinery, the protein synthesizing machinery, to pick the correct amino acid from the pool of other amino acid is amino acyl tRNA synthetase. Once that is done, this amino acid then has to be attached to the 3' end of our tRNA, which referred to as the acceptor arm. This same enzyme, amino acyl tRNA synthetase, catalyzes the linking of that selected amino acid to the tRNA before this tRNA is brought into the ribosome to form that complementary base pairing with mRNA. This process itself is going to lead to an activated tRNA, a tRNA that has got the correct amino acid linked to its 3' end. Like I said, the enzyme that catalyzes this process, including identifying or picking the amino acid from the cytoplasm, don't forget, is amino acyl tRNA synthetase. Let's look at the first process, just to simplify what I'm trying to say there. So what you see here is amino acyl tRNA formation. We want to find out how is this amino acid, selected. So the first thing that happens is that we have the amino acid. If you remember our lectures on proteins, this is an amino acid. They will have these 3 regions are going to be the same, but they differ at the ARA group. This amino acid is going to react with ATP to form a high energy molecule, like an input of energy. This reaction will be catalyzed by amino acyl tRNA synthetase. And what will happen is that we're going to form an amino acyl adenylate. That is to say, you have your amino acid and you've added 1 phosphate to it, forming a high energy bond, isn't it? And of course, you're going to have 2 phosphate groups released because this was ATP with 3 phosphate. And at the end of the day, you've utilized 1 phosphate by binding it to amino acid. You have 2 being released. At this point then, this amino acid can now be taken to its corresponding tRNA. And what I mean by corresponding tRNA is that the amino acid that's going to bind on the S side, on the 3' end of the tRNA, must be the one that is going to enable complementary base pairing between the anticodon and the codon. So once this is done, we're now going to have your amino acid adenylate now binding to the 3' end of your tRNA. So what you see here now is the charging of the amino acid. Now, keep in mind, the charging of an amino acid first has to start with selection of that amino acid. And at this point, the amino acid tRNA synthetase has already done that for us. So this time around, you want to bring this charged amino acid and link it, covalent link it using an ester bond to the 3' end of a tRNA, which is already marked like we said in the previous slide. So a charged tRNA molecule, or should I say to charge this tRNA, again, the same enzyme is used to catalyze this reaction, which is amino acid tRNA synthetase. This same enzyme is the one that activated the amino acid by reacting the amino acid with the ATP, forming a high energy molecule amino acid and a charged amino acid. The charged amino acid in that state can now be attached to tRNA. The same enzyme comes in to catalyze that reaction. At the end of this process, we have a charged tRNA. What do we mean by charged tRNA? A charged tRNA is a tRNA that is carrying an amino acid on its 3' end, which corresponds to the anticodon. By corresponding to the anticodon, meaning that the information on the anticodon can form complementary base pairing with the information on the codon on the mRNA in the ribosome, of course. Okay, now, how many amino acid tRNA synthetases should we have? So think about it. Why am I asking this question? We are saying that at each point, an amino acid has to be selected from the pool of other amino acids in the cytoplasm. We have 20 amino acids that naturally exist, isn't it? And the mRNA, we can assume at this point, is carrying different combinations of codons that are going to give us different amino acids. It's not always the case. I'm just giving you a scenario where you have your mRNA carrying 10 codons, and each one of those 10 codons is going to give rise to a different amino acid. At this point, we're assuming there's no stop codon. I want to find out how many amino acid tRNA synthetases should we have? You know, the good part about online classes is that everyone here can be sleeping, or you can even log out, since I'm not able to see you. So it's very difficult. That is why you need to come back to, to those lecture theaters, because they are not allow you to sleep, right? In fact, I've missed you guys. I was hoping you'll be coming back very soon, but anyway, let's see. You might be coming back very soon. So I've given you a scenario. I've given you an mRNA molecule, and I've told you this mRNA molecule has got 10 codons. 10 codons. And those codons, none of them is specifying a stop codon, meaning that each one of them is giving us, in this scenario, a different amino acid. So then, how many amino acid tRNA synthetases should you have? Just unmute yourself and give it a try.
Madam, can I give it a try?
Of course.
Approximately 20 so.
So for the scenario that we have, why should there be 20? Maybe the understanding is even what's even more important. How did you come up with 20? Did you just guess? Is it a lucky guess? Or it's just one of those things?
Just guessed.
You just guessed. It's a good guess. Sometimes even guessing can help you, but like I always tell you guys, when you write your test, please do not guess. We don't guess there. We always have to know what we're talking about. It's a good guess though, but I want someone to tell me, okay? We have 20 amino acids that naturally occur in our living systems, and we're trying to say that what the cell is going to do before it can translate, interpret the information on the mRNA is to select what is sitting on the mRNA. So for example, in the ribosome, there is an AUG. This AUG should be translated to the amino acid methionine, meaning that the tRNA that should come into the ribosome should be the one that is carrying what? Methionine attached to it, isn't it? So the question requires you to think like that. How many amino acid tRNA synthetases, should we have? Because we know the selection of those amino acids is, aided by this enzyme. Remember, it is the one that activates that amino acid. By adding a phosphate to it, it is marked for its attachment to a tRNA. That is like activating it. So then if all of them, if there's only one amino acid tRNA synthetase, how is it going to select all those 20 amino acids? Because I think it can make a mistake. I don't know if you're thinking in those lines. Okay, what we're trying to say here is that we must have approximately 20 amino acid tRNA synthetases to distinguish the 20 amino acids that we have that naturally occur in our cytoplasm. This time around, when I say cytoplasm, of course, we know it can be a bacteria because translation in both cells takes place in the cytoplasm. So we should have approximately 20 amino acid tRNA synthetases based on what? The fact that, we have 20 amino acids and each one of them has to be selected, brought to the tRNA, and then finally this tRNA can be charged and taken into the ribosome to complementary base pair with a specific, with its exact, codon to form that anticodon-codon relationship. So, okay, these are just notes for you to read, and these are just examples. Cells contain many tRNAs, of course, because we know that the third position on the codon is wobbly, isn't it? So you can have more than one tRNA, binding to the same codon, because the third position on the codon sometimes can mismatch but still give you the same amino acid. So what I'm trying to say here is that the question we asked, how many amino acid synthetases do we have? We have got approximately 20, one for each of the 20 naturally occurring amino acids, because we know this enzyme is the one that recognizes these amino acids. So a single amino acid synthetase can recognize multiple tRNAs the same. Okay, this is where my battery wants to go to sleep. Okay, let me just try to see how far I can go. Okay, guys, now I want us to understand, how is it possible that one tRNA, has it already cut? Can you hear me? Not yet. Okay, I'll end on this slide. We can, I think, continue tomorrow. Let me just explain this. So here we want to understand. We said that the genetic code, one of the things that it exhibits is that it is degenerate. You can have one codon, you can have one amino acid being specified or coded for by more than one codon. But then one tRNA also can recognize more than one codon because of that base pair on the third position on the codon. It is, we say it is wobbly, meaning it can allow for a mismatch as long as the first two positions are, the same, complementary with the tRNA. So what we're trying to say here is that one tRNA can form complementary base pairing with more than one codon, specifying the same amino acid though. Like what you can see here, phenylalanine. If on the mRNA you have UUC, the tRNA that comes to the ribosome must be carrying a phenylalanine. But we can also have the same tRNA coming in when the codon is UUU. Why? Because the first two positions must match for this to happen. The third position can allow for a mismatch, because we know here a G, for example, a U here should best pair with an A, but that's not the case. So meaning that this position does not need to be exact. As long as the first two positions can form stable complementary base pairing, the third one can mismatch, but still allow for the same tRNA to recognize that codon. So in short, each tRNA is specific to only one amino acid. Okay, each tRNA is specific to only one amino acid. For example, this tRNA is for phenylalanine, but it can recognize more than one codon as long as the first two positions of that codon are the same. The third one, because of the wobble nature of the genetic code, can allow for a mismatch. Ladies and gentlemen, allow me to end here. We can start from here tomorrow at 8 hours. For me, have a good day and take care. Bye for now. Guys,